Imagine having like a secret instruction manual inside you, one that dictates everything your hair color, maybe even your risk for certain diseases. Well, you do. It's your genetic code. And today, we're diving deep into that manual specifically for you, our future nursing experts. Welcome to the deep dive. We take complex info and make it clear, concise, and hopefully captivating. Our mission today: unpacking genetics and genomics from Lewis's MedSurg Nursing 12th edition.
Exactly. This deep dives, it's really designed to equip you, not just for exams or the NCLEX, but maybe more importantly, for understanding the real shifts happening in patient care. We'll go step by step through the fundamentals, mutations, those inheritance patterns, diagnostic tools, and of course, the crucial nursing management piece.
So, what's the big thing today? It's actually pretty simple, but profound. Genetics and genomics, they're not just abstract science anymore. They're actively changing healthcare delivery, and nurses like you are right at the center of it all.
Okay, let's unpack this then. We often hear genetics and genomics used, well, almost interchangeably, but there's a key difference, isn't there? Why is getting that distinction right more than just, semantics for nurses?
That's a really great starting point. So, genetics, think of it as studying individual genes and how they influence inheritance. It's like focusing on just one chapter, maybe one specific instruction in that big manual to understand a single trait or a specific condition, how it gets passed down.
Okay, so genetics is the specific page. What about when we zoom out, look at the whole book? That's genomics, right? How does that help with something complex like heart disease? It's exactly right. Genomics takes that broader view. It studies all of a person's genes, the entire genome, and crucially, how those genes interact with each other and with the environment. This helps us tackle complex diseases, heart disease, diabetes, most cancers. These usually aren't just one rogue gene. It's often multiple genes working together, plus lifestyle, environment, all of it. It helps explain those situations, where someone lives really healthy but still gets sick, or vice versa.
Right, so these are the fundamental building blocks we're talking about. Genes made of DNA, this amazing molecule storing all our information. And it's wild that pretty much every single body cell, every somatic cell, has the same DNA blueprint.
It is. And how that information is stored is just fascinating. DNA uses four chemical bases, adenine, guanine, cytosine, and thymine, A, G, C, T. They pair up very specifically. A always with T, and C always with G. These pairs form the rungs of that famous double helix structure, like a twisted ladder. And this structure isn't just neat, it's functional. It allows DNA to replicate itself almost perfectly, so when cells divide, each new cell gets a complete, accurate copy. That's life.
So DNA holds the master plan, but how does the cell actually do anything with it? How does the plan turn into action?
That's where RNA comes in. Think of RNA as the messenger. It's similar to DNA, single-stranded though, and it uses uracil, U, instead of thymine, T. Its main job is to ferry the genetic instructions from the DNA, which stays safe in the nucleus, out to the parts of the cell that actually build proteins. This whole process, protein synthesis, happens in two main steps. First, transcription, making a messenger RNA, or mRNA, copy from the DNA template. Then, translation. The mRNA hooks up with a ribosome, and another type of RNA, transfer RNA or tRNA, brings the right amino acids in the right order to build the specific protein coded by the mRNA. Proteins do all the work.
And speaking of cells dividing,
Right.
quick clarification for everyone listening. Mitosis, that's your everyday cell division, right? Making identical copies for growth or repair.
Correct. Exact replicas.
But meiosis is different.
Very different. Meiosis only happens in the germline cells, the ones that become eggs or sperm. It produces four unique sex cells, each with only half the chromosomes. Absolutely essential for sexual reproduction and inheritance.
Okay, this is where it gets, well, really interesting for clinical practice. What happens when that super precise genetic blueprint gets a typo? A change?
That typo is what we call a mutation. It's any change in the usual DNA sequence. It could be tiny, a single base swapped, or huge, like a whole chunk of a chromosome missing or moved. These changes can alter the protein that's made maybe the wrong type, maybe too much or too little, and sometimes that leads to disease. A classic example is sickle cell disease. A single tiny change, one DNA base, thymine, replacing adenine in the beta-globin gene, results in abnormal hemoglobin. You've probably seen the pictures. It makes red blood cells stiffen and form that sickle shape. That causes all sorts of problems with blood flow and oxygen delivery.
And these mutations, they don't just appear out of nowhere, right? There are different ways they happen.
Right, two main categories. First, germline mutations. These are inherited from a parent. They were present in the egg or sperm, so they end up in basically every cell of the person's body, right from the start. Then you have acquired or somatic mutations. These happen during a person's lifetime in a specific cell. Maybe a mistake during DNA copying or damage from things like UV radiation from the sun causing skin cancer.
Or smoking causing lung cancer.
Exactly. Those toxins are mutations. Now, these acquired mutations are passed on to all of the cells that descend from that one mutated cell, but importantly, they are not passed on to the next generation. We actually have DNA repair systems that fix most errors, but they can become less efficient as we age. So mutations can accumulate.
And these changes, the inherited ones especially, they can be passed down in families in predictable ways. Understanding that is so key for risk assessment.
Absolutely key. We talk about inheritance patterns. One common type is autosomal dominant. This is caused by a mutation in just one copy of a gene pair on an autosome. Those are the non-sex chromosomes. The dominant mutated allele wins out. These disorders affect males and females equally, and you often see them in every generation. But here's the tricky part for nurses. They can show variable expressivity.
Meaning the symptoms can be really different from person to person, even in the same family?
Precisely. Some might be severely affected, others mildly. And they can also show incomplete penetrance, meaning someone can have the mutation, but maybe show no signs of the disease at all. Yet they can still pass it on. Think of conditions like Huntington disease or the BRCA gene mutations linked to breast and ovarian cancer risk.
Okay, so that's dominant. What about recessive?
Autosomal recessive disorders are different. You need two copies of the mutated gene, one from each parent, on an autosome to actually have the disorder. If you only inherit one copy, you're a carrier. You usually don't have the condition yourself, but you can pass that mutated gene to your children.
So carriers are usually asymptomatic.
Typically, yes. This is why recessive conditions often seem to skip generations. They only appear when, by chance, both parents are carriers. Think cystic fibrosis, sickle cell disease again, Tay-Sachs.
And then there are the ones linked to sex chromosomes?
Right. X-linked recessive disorders. The mutation is on the X chromosome. Since males only have one X, they are usually much more severely affected if they inherit the mutated gene. Females have two X chromosomes, so the normal copy on their other X can often compensate. They tend to be carriers, often without symptoms. Affected fathers pass the mutated X to all their daughters, making them carriers, but not to their sons. Think of hemophilia or Duchenne muscular dystrophy.
And Y-linked. That seems straightforward.
It is. Y chromosomal inheritance only affects males and is passed directly from father to all his sons.
Connecting this back, the Human Genome Project, that was huge, right? Finished back in 2003. How did that really change things?
Oh, monumentally.
Yeah.
It mapped the entire human genome. That knowledge base is transforming everything. It helps us diagnose diseases better, detect genetic predispositions much earlier, it assists in risk assessment, even helps match organ donors more effectively. It really paved the way for personalized medicine.
And when we talk about genetic disorders, they aren't all the same type of problem, are they? How do we generally classify them?
Good point. We can think about them based on the type of DNA change. First, single gene disorders. Like the name suggests, a mutation in just one gene. Cystic fibrosis, sickle cell, polycystic kidney disease, these are examples. They're often quite impactful, but actually relatively rare overall.
Okay, what's more common then?
Much more common are multifactorial inherited conditions. These are the complex diseases, heart disease, type 2 diabetes, most cancers. They result from variations in multiple genes interacting with environmental factors and lifestyle choices. It's that complex interplay.
Like the BRCA example, inheriting the gene mutation significantly raises cancer risk, but environment and other genes still play a role.
Exactly. It's not destiny, but it's a major risk factor influenced by other things.
Then there's this fascinating field, epigenetics. What's the deal there? It's not changing the DNA code itself.
Correct. Epigenetics studies changes in gene expression, how genes are turned on or off, that don't involve altering the underlying DNA sequence. These epigenetic marks can be inherited during cell division and can be influenced by things like age, diet, exercise, stress, environmental exposures. It helps explain why even identical twins with the exact same genes might develop different diseases or show different traits over time. Their life experiences create different epigenetic patterns.
Wow. Okay, and the last category.
Chromosome disorders. These involve larger scale changes. Problems with the structure of chromosomes, or having too many or too few copies of entire chromosomes or large segments. The classic example is Down syndrome, trisomy 21, caused by an extra copy of chromosome 21. Another important one in hematology is chronic myelocytic leukemia, or CML. It's often caused by a specific chromosomal translocation called the Philadelphia chromosome, where bits of chromosome 9 and 22 swap places.
Okay, so we've got the basics, the variations, the classifications. Now, the really practical stuff for nurses. How do we use this knowledge clinically? Let's talk screening and testing.
Right. And first, it's important to distinguish genetic screening from genetic testing. They sound similar, but the purpose is different. Screening is usually offered to a broader population, maybe a specific group considered at risk but who don't have symptoms or a strong family history yet. Think of the ifetoprotein or AFP screening offered during pregnancy to look for potential fetal issues.
Okay, so screening is broad, looking for potential risk. What's testing then?
Genetic testing is more targeted. It's done for individuals or families who have a specific reason. Maybe they have symptoms suggesting a genetic condition or a known family history placing them at higher risk. The test analyzes specific chromosomes, genes, or gene products, looking for a particular mutation or predisposition. Samples can be blood, saliva, skin cells, even prenatal samples like amniotic fluid.
And the reasons for testing are really diverse, right? Can you walk us through some of the main types?
Sure, there's carrier screening, like we mentioned, to see if someone carries one copy of a gene for recessive disorder like CF or sickle cell. Diagnostic testing is used to confirm or rule out a condition when someone already has symptoms, maybe testing for Huntington disease if symptoms appear. Newborn screening is huge, done routinely in hospitals to catch things like PKU or congenital hypothyroidism early so treatment can start immediately.
What about for medications?
That's pharmacogenomic testing. It looks for genetic variations that affect how someone metabolizes or responds to drugs. Super important for drugs like warfarin, where the right dose can vary widely based on genes.
And testing for future risk.
Yes, that includes predictive testing, which identifies mutations like BRCA12 that increase future risk, allowing for preventative strategies like increased surveillance or even prophylactic surgery. And pre-symptomatic testing, which is for people with a family history of a late-onset disorder, like Huntington's, to see if they inherited the mutation before any symptoms show up.
Plus prenatal testing during pregnancy.
Right, prenatal diagnostic testing, like amniocentesis or CVS, looks for genetic changes in the fetus itself.
Interpreting these results, though, it's not always black and white, is it?
Definitely not. A positive result means the lab found the genetic change they were looking for. It might confirm a diagnosis, identify carrier status, or indicate increased risk. But, and this is crucial, a positive predictive test often can't tell you for sure if or when someone will develop the disorder or how severe it might be.
Right, like the ApoE4 gene for Alzheimer's risk, having it increases risk but doesn't guarantee you'll get the disease.
Exactly. And a negative result means the lab didn't find the specific alteration tested for. Usually that means the person isn't affected, isn't a carrier, or isn't at increased risk for that specific mutation. However, the test might have limitations. It might have missed a different mutation causing the same disease, or maybe the gene responsible isn't even known yet. So a negative result isn't always a clean bill of health.
And then there are those direct-to-consumer tests, the DTC ones you see advertised. People swab their cheek at home, mail it in.
Yes, and they get results online or by mail. While they can be interesting, they come with significant risks. People might misunderstand the results, especially risk estimates. They might be misled by claims that aren't scientifically validated. They can make major health decisions without talking to a doctor or genetic counselor.
And privacy concerns, too.
Big privacy concerns. As nurses, it's really important to educate patients about the limitations of DTC tests and strongly advise them to discuss any results, especially medically relevant ones, with a healthcare professional. Don't make decisions based solely on a DTC test report.
This all leads into the ethical side of things, which is huge in genetics.
Absolutely huge. Privacy, potential discrimination, these are major concerns. Thankfully, we have the Genetic Information Non-Discrimination Act, or GINA. It's a U.S. federal law that prohibits health insurers and employers from discriminating against individuals based on their genetic information. It prevents them from using genetic test results to deny coverage, raise premiums, or make hiring or firing decisions.
That's incredibly important protection. But GINA doesn't cover everything, like life insurance or long-term care insurance, right? And there are other ethical wrinkles.
Correct. It has limitations. And other issues come up, like finding out information about relatives who didn't consent to testing, or uncovering unexpected family relationships, like non-paternity. These situations require incredibly sensitive handling.
Okay, moving beyond diagnosis. We're now using genetics in some amazing ways for treatment and identification. Let's talk tech.
For sure. DNA fingerprinting, for example. Using PCR, polymerase chain reaction, we can amplify tiny amounts of DNA, make millions of copies. It's fundamental in forensics identifying suspects or victims. Also used in paternity testing, and it's a key confirmatory test for HIV, especially in newborns of HIV-positive mothers.
What about those DNA chips you hear about?
DNA microarrays, or DNA chips. These let researchers look at thousands of genes simultaneously to see which ones are active or inactive in a particular cell type or under certain conditions. They can help identify specific mutations or gene expression patterns linked to disease, or even predict how effective a drug might be for someone. Basically, tiny spots of known DNA sequences are on a chip. You apply the patient's labeled DNA and see where it sticks, revealing patterns.
And mapping disease associations across populations.
That's where genome-wide association studies, or GWOs, come in. They scan the entire genomes of large groups of people, comparing those with a disease to those without it. The goal is to find common genetic variations that are associated with an increased risk for specific diseases, especially those complex multifactorial ones. GW's findings are really driving the development of personalized medicine.
Which ties directly into pharmacogenomics and pharmacogenetics, right? Understanding why people react so differently to the same drug.
Exactly. Genetics plays a huge role in drug response. Pharmacogenomics is the broader study of how the entire genome influences drug effects. Pharmacogenetics is a bit narrower, focusing on how variations in single genes affect drug response. The goal is the same: personalized medicine, getting the right drug at the right dose to the right person based partly on their genetic makeup.
Can you give us a key example nurses might see?
A classic one is the cytochrome P450 CYP450 enzyme system in the liver. These enzymes metabolize a huge number of common drugs. Genetic variations can make these enzymes work slower or faster than usual. Slow metabolizers might need lower doses to avoid side effects, while fast metabolizers might need higher doses for the drug to be effective. We see this with the blood thinner warfarin. Testing for variations in the CYP2C9 and VKORC1 genes helps predict the optimal starting dose, improving safety. There are many other examples tests related to clopidogrel, certain cancer drugs like trastuzumab.
And then gene therapy. This sounds like science fiction, but it's actually happening.
It is. Gene therapy is still largely experimental, but it aims to treat disease by correcting the underlying genetic problem. Usually, this involves using a carrier, called a vector, often a modified virus, that can't cause disease to deliver a functional copy of a gene into the patient's cells. The idea is to replace a missing gene or provide a working copy of a faulty one. It's complex, there are risks, but the FDA has approved gene therapies for a few specific conditions, like a type of inherited blindness and certain pediatric leukemias. It's a really exciting frontier.
Alongside that, stem cell therapy. What are stem cells again?
Stem cells are unique, unspecialized cells. They have this remarkable potential to either remain as stem cells, dividing to make more, or to differentiate to develop into various specialized cell types like muscle cells, blood cells, nerve cells.
And we have them as adults.
Yes, adult stem cells exist in many tissues: bone marrow, brain, skin, muscle. Their main job seems to be maintaining and repairing the tissues where they're found. We already use stem cell therapy in some areas, like bone marrow transplants, hematopoietic stem cell transplantation, for cancers, treating severe burns, some orthopedic procedures. And research is intense, exploring how we might use them to repair or replace tissues damaged by disease or injury. Think heart disease, Parkinson's, diabetes. Still lots to learn, but huge potential. So with all this incredible science, it really brings us back to the core question: how do we as nurses actually integrate this into providing compassionate, effective patient care?
Yeah, it seems like the nurse's understanding is just paramount. You need to grasp these concepts to help patients and families navigate really tough decisions about genetic issues, right? You're often the one connecting them to resources, explaining things.
Absolutely.
Yeah.
And you don't do it alone. Collaboration is essential. Working with specialized genetics nurses, GCNs or APNGs, and genetics counselors is key. They have specific training in genetics and, importantly, in counseling skills to help patients deal with the complex emotional, ethical, and social issues that often come with genetic information. Worries about insurance, career, marriage, having children.
Let's make it real with a scenario. Say you have a 30-year-old pregnant patient. Her youngest child has cystic fibrosis. Now she's unexpectedly pregnant again and very anxious. Her husband asks about the odds for this baby. She's considering fetal testing. What's the nurse's role?
Okay, multifaceted role here. First, provide accurate information about the testing options available. But, critically, emphasize the need for formal genetic counseling. You might use tools like a Punnett square to visually explain the probabilities. Since CF is autosomal recessive, if both parents are carriers, you'd show them the 1 in 4 or 25% chance with each pregnancy of having an affected child. You'd also help construct a family pedigree to visualize the history. But beyond the science, you address the psychosocial aspects, privacy, confidentiality, the emotional weight of potential decisions, like possibly terminating the pregnancy. Your role is support and information, facilitating their informed decision-making, not directing it.
Okay, another one. A woman with a strong family history of breast cancer gets diagnosed herself. She asked us if she and her daughters should get tested for BRCA mutations.
Again, start with education. Discuss the pros and cons of testing. What could the information tell them? What are the limitations? What are the emotional impacts? And definitely bring up GINA. Explain clearly how this law protects them against discrimination from health insurers and employers based on genetic test results. Knowing their rights is empowering.
And this all highlights the importance of a good family history, doesn't it? That's basic nursing assessment, but maybe even more critical now.
Absolutely fundamental. And not just asking, does anyone have heart disease? We need a comprehensive family health history. Ideally, creating a pedigree chart covering at least 3 generations. You need details. Who had what condition, at what age were they diagnosed, cause of death if applicable, their relationships, ethnic background. You're looking for red flags. Disease in multiple close relatives. Disease occurring at an unusually early age, like a heart attack before 35. A condition appearing in the less expected gender, like male breast cancer. Certain combinations of diseases like breast and ovarian cancer, or heart disease and diabetes clustering in the family.
And if you see those red flags,
That's a trigger for referral. These patients need further evaluation, possibly genetic counseling and testing. Your thorough assessment is the first critical step.
so it really loops back to patient education and health promotion, using these tools, the history, the pedigree, maybe Punnett squares, to explain risk clearly.
Exactly. And then tailoring health promotion based on that risk. If someone has familial hypercholesterolemia confirmed, you focus on lipid monitoring and management. If they have a strong family history of type 2 diabetes, you emphasize lifestyle modifications even more strongly. And always, always maintaining confidentiality, respecting their values and decisions throughout the process. You are their advocate and guide.
Wow. We have definitely covered a massive amount of ground today, from, this tiny DNA-based pairs all the way up to the biggest ethical questions in healthcare, a real deep dive into our genetic blueprint and what it means for nursing.
It really is foundational. So to quickly recap the key takeaways for you as you move forward in your nursing careers. First, understanding the difference between genetics and genomics. It's not just terminology. It shapes how you assess patient risk. Second, recognizing those inheritance patterns helps you anticipate disease risk and guide crucial family conversations. Third, genetic testing is powerful, yes, but interpreting results needs real care, understanding limitations, and expert counseling is often essential. And maybe most importantly, your role as a nurse is absolutely central. You're the educator, the supporter, the advocate, navigating patients through this complex, rapidly evolving genetic landscape, personalized medicine, gene therapy. The future relies on nurses having this knowledge.
That's a great summary. So, thinking about that future, considering how fast genetic science is moving, what new ethical challenges do you see coming up for nurses in the next, say, 10 years? And maybe how can our listeners, as future nurses, start preparing to face them? Something to think about. Thank you so much for joining us on this deep dive into genetics. We really hope this has clarified things and maybe sparked some more curiosity.
It's truly been a pleasure exploring this vital topic with you all. Keep learning, definitely keep asking questions, and know you're going to make a real difference.
Until next time, on The Deep Dive.